Welcome to the Curiox Biosystems webinar. We have 3 speakers today, 2 from the National University of Singapore, Dr. Joanna Brzostek, Senior Research Fellow, and Chen Elijah, postgraduate student, both from the lab of Dr. Nicholas Gascoigne. In addition, Dr. Charles Martin, who is Director of Marketing at Curiox Biosystems. The presentation will last approximately 30 minutes. Please type your questions in the question box, and they will be addressed at the end of the session. A recording of this webinar will be available shortly after the presentation at www.curiox.com. Now, may I introduce Dr. Charles Martin.
Thanks, Sheila, and welcome everybody to our webinar. So Joanna and Elijah will follow my short presentation and will share experience and data from their T-cell screening work. First, I'm just going to give a brief overview of the drop array system. So let's see which step in the flow cytometry workflow causes challenges. And a simple answer to that question is every step. And this slide shows a brief overview of a flow assay workflow. And there are many possible areas of variation. The way cells are handled, particularly washing, the setting of instrument parameters, and how the population of cells are gated. Any one variation in the workflow will cause changes in the data, potentially leading to a different experimental outcome. So there has been tremendous effort in the flow community to standardize a workflow and produce robust flow data that can be compared to each other independent of operator, time, and location. Here's a summary of a recent review paper discussing the standardization of flow cytometry to achieve consistency and reproducibility and thereby enhance quantitation. From the previous slide workflow, we learned that there are 3 major components. Data analysis, oops, let's see here. Sorry, lost my train of thought. Okay. So there's 3 major components: data analysis, the flow cytometer, and sample prep. Unfortunately, we've achieved tremendous progress in 2 out of the 3 areas. The most challenging component is sample prep. And in this particular review paper that's cited down at the bottom left, the authors claim that we have to apply much more effort for our operators to strictly follow SOPs or protocols and to ensure they minimize mistakes and report deviations. All experts in the field do not agree. They firmly believe pushing operators is not necessarily the best solution. We need a different solution if we want to achieve robust data on flow cytometry. And automation is the solution, many say, to resolving the challenge of reproducibility. And in this slide, I'm showing statements from several leading experts in biopharma. So Mark Ettinger is the head of the flow business at Q2, which is a major CRO globally. And I'm going to let we'll send you these slides after the presentation so you can read these at your leisure. And then also we have Dr. Sadiq Kasim, who's now CSO at Mustang Bio, and he formerly at Novartis, where this quote is from, commenting on automation addressing the challenges. So at this point, you may think, okay, automation is really important in improving flow cytometry data, but I'm not ready to embrace a new technology. I want to automate what I'm familiar with, the centrifuge. And maybe you think you can build an inexpensive automation using the centrifuge. And you're not certainly the first to come up with that idea. So in this slide, I'm comparing the instruments needed for building automation. And so we have the DA cell on one side, and then you can see the chart at the bottom where it's comparing what's needed for automation. And when we use DA cell, we need only 4 tools: a workstation, an 8-channel pipetting head, or maybe even a 4-channel pipetting head, a gripper to move the plates around, and the DA cell washer. That's it. Whereas in contrast, the centrifuge-based automation requires additionally a 96-channel head, a plate washer, and an automation-friendly centrifuge. Another important component is that centrifuge-based automation requires expert integration and maintenance. When we have these 2 different automations, the DA cell can finish 3 centrifugation equivalent washes within 7 minutes, while centrifuge automation takes 30 minutes. And the increased time with the centrifuge is due to about 6 times more movements required for the centrifuge-based washing. So overall, DA cell washer automation can be realized at a significantly lower price and take less than half the bench space than a centrifuge-based one. So the drop array cell washer is positioned to minimize all of the challenges listed under the centrifugation of cells. In a conventional wash process, most people use a centrifuge and a multi-channel pipette, and a wash of 2 centrifugations takes a total of 15 to 20 minutes, depending on what your particular protocol is. In contrast, the DA cell washer takes care of the entire wash process in approximately 2 minutes without centrifugation. And also without centrifugation, cells do not experience any external forces or stress. And if you have low cell numbers, which may be difficult to pellet with a centrifuge, you don't have that problem with the DA cell washer. So now in this slide, I'm going to show a short movie clip of how the DA cell washer runs. And in the movie, there's 2 wells that have droplets of green fluorescent polystyrene beads down here. And then right here, there's 2 wells full of food coloring or dye that's taking the place of labeled antibodies. And then the polystyrene beads represent cells. So what happens is typically in an assay, you'd add your antibodies and to your cells, and then they would have about 20 minutes or so to incubate, and that's what the time that's allowed for the cells to settle. So after the cells have settled, the user just places the plate in the washer and hits the start button. The plate goes in and the fluidics head comes down to the plate. So in this video, we've lifted the cover to show the movements of the head and the plate inside. The head has 192 nozzles total. So there's 2 nozzles per well, 1 for dispensing in each well and 1 for aspiration in each well. And during the 2-minute operation, it performs 7 cycles each of dispensing and aspiration. So when the wash is complete, as we show here, and the plate comes out, you can see the food coloring solution is completely gone in these 2 wells right here. So the food coloring again represented the tagged antibodies, while the fluorescent green beads in these 2 wells represent cells, and they were retained in place. And so the wells that contained the food coloring now look just like these other wells, which contain just buffer. And you may wonder, well, how come the food coloring is washed away while the beads are retained? And just to clarify, the beads or cells are not attached to the plate by any actions. They're simply sitting on the plate by gravity without any adhesion. And I'm going to show you in the next slide how this works with this little cartoon here. So the retention of beads and cells on the plate is based on well-characterized forces of gravity and laminar flow, which are very well-characterized natural forces. The secret is that the use of each force is on an almost flat, wall-less plate, so we can control the forces, unlike a standard plate with walls where the forces would be uncontrollable. At the bottom of the slide, you can see the working mechanism of the cell washing by DA cell. So down here, the cells are mixed and settled on the plate by gravity, and the settling takes 20 minutes or so, usually during standard incubation periods. And once the cells are settled, the action of dispensing and aspiration above the cells does not disturb them. The wash solution forms a laminar flow during dispensing and aspiration by the nozzles I described earlier, and minimizes turbulence of the liquid flowing over the settled cells by laminar flow. Now, the velocity of the fluid when it approaches the surface of the plate and above the cells approaches 0. And so over that thin fluidic layer, washing happens by diffusion rather than turbulence of the fluid. And that's how we can wash the cells without losing them. So when we perform identical dispensing and aspiration in a conventional walled plate, that action of dispensing and aspiration causes a lot of turbulence, which will lift the cells from the bottom of the plate and lead to cell loss. So in the next couple of slides, I just want to quickly highlight some data we've run with collaborators and customers, and it will complement the data that Joanna will present. So again, these next couple of examples show optimized DA cell methods. So every time we work with a customer, we optimize to their specific protocols. In this case, I'm going to show how the method can retain more cells, particularly these splenocyte samples. So now the flow rate of dispensing and aspiration can be adjusted, just as the time can be adjusted, and we've optimized for this particular assay. So here we show enhanced cell retention during a FOXP3 staining of splenocytes. And again, this is a relative comparison between the 2 methods. So you can see the chart legend here. We've got 2 methods, 2 flow rates with a DA cell washer, and then washing with a tube and a centrifuge. And the maroon colored bars and the maroon and gray checkered ones are the DA cell washer at 2 different flow rates. So you can see that both methods with the DA cell have better retention than the tube method. And again, this is optimized for this particular lab's protocol. And this slide also shows enhanced retention with the same color scheme as before, with the maroon being the DA cell method and the gray being the tube. And in this case, we have better retention of TILs, or tumor infiltrating lymphocytes. And again, this is relative compared to the tube method. So the tube method is ratio of 1, and then you can see there is greater cell retention with the DA cell washer. And lastly, I just want to talk quickly about reading the plates on a flow cytometer. So the DA cell plate can hold up to 70 microliters in its standard configuration. And that's great for washing assays, but it might be too small to read the plate directly on a flow cytometer. It depends on your flow cytometer. And often a flow cytometer, depending on its behavior, how it aspirates the cells up into the fluidic system, it might require 100 to 150 microliters of starting fluid. So we do have an accessory to increase the volume of the plate when it's time to read. Now, for example, in this case, it's on an HTS plate reader on a Becton-Dickinson flow cytometer. So the graph on the left shows the comparison between a conventional U-bottom plate and the drop array plate. And again, we're following the same color scheme here. The gray bars are U-bottom, and the maroon bars are the drop array plate. And so as the graph shows, the reading from the drop array plate is just as good as reading from your standard U-bottom plate. Even when the cell numbers are as low as 10,000, the recovery of cells by direct reading are comparable between U-bottom and a drop array. And I'm going to turn it over to Joanna now and Elijah.
Fantastic. Good morning. So I'm going to tell you something about how we use the DA cell washer to look at T cell activation, both looking at thymocytes and peripheral T cells. So as a lab, we are interested in understanding how the TCR affinity to peptide MHC can regulate T cell development in the thymus and the peripheral T cell effector functions. So if you look at the TCR peptide MHC affinity, the high affinity interactions lead to cell death by negative selection during the thymic development, whereas in the peripheral T cells, the same high affinity induces proliferation and effector functions. And in the peripheral T cells, there is a very critical role for IL-2 signaling in this process. However, the low affinity interactions, which in thymocytes give rise to positive selection, do not induce any effector functions in the peripheral T cells, but those are required for survival and functional responsiveness, as well as homeostatic proliferation in the peripheral T cells. And in all those processes, there is actually a critical role for IL-7 signaling. So as you can imagine, being immunology lab, we do a lot of flow cytometry. And like a lot of flow cytometry labs, we encounter some very common problems. So some of which are related to looking at the TCR signaling in the thymus, and some which are more specific to looking at cytokine signaling in mature T cells. So one of our problems, which is very common with flow cytometry, is trying to gauge on the correct populations, which would correspond to the different thymocyte developmental stages. And one of the problems is that some of our critical antigens have very low cell surface expression. So it's actually quite difficult to gauge on the relevant populations. Another challenge that we encounter, and Elijah is going to talk about this later, we try to develop an high throughput inhibitor screen to identify new mediators of TCR signaling. And for this, we need a very consistent and time-efficient high throughput thymocyte activation assay. Whereas for our experiments looking at cytokine signaling in mature T cells, one of the challenges we encountered was very problematic antibody staining with some of the antibodies. And another problem we encountered was we tried to develop an experimental system that will allow us to actually quantify the duration of a cytokine signal. And for this, we need a very fast washing procedure to examine how long will the signal persist after the cytokine has been removed. So I just want to share with you how at least some of those problems got solved by using the D8 plate washer. So starting with the thymocyte analysis, looking at very conventional thymocyte subset analysis is we are looking at the double positive and the single positive development just by looking at the expression of the CD4 and CD8 co-receptor. And I think it's pretty immediately obvious that using exactly the same cells and exactly the same antibodies, we are getting higher MFI values for both our CD4, as you can see here, and specifically for our CD8 antibody, which allows us to gauge our populations better. And this is going to be even more striking if we look at the very critical cell surface markers, which are normally expressed at much lower levels in thymocyte. So for example, if you look at the TCR and the CD69 plots, which those plots are really critical because they allow us to examine different stages in thymocyte development, I think it's quite obvious that with the D8 washer sample prep, we are actually getting higher MFI values, as you can see here, both for the CD69 and for the TCR, and that actually allows us to gauge our populations better. And I think this would be very critical for anybody who tries to actually use automated gating. So to continue the thymocyte story, I'm going to hand over to Elijah, who is going to share with you more about his inhibitor screen assay.
Thank you, Jo. I hope I'm audible.
Yes.
Yeah. Okay. So, good morning. I'm Elijah, and I'll be introducing an assay that we have developed in our lab for the identification of modulators of T cell signaling and T cell activation. Our essay has been published or is being published, and the details can be found at the bottom of the slide. Our essay is based on the physiological process of thymocytes undergoing a selection process in the thymus, in which they undergo a caspase-dependent apoptosis, where there is a strong interaction between the TCR and the self-peptide MHC ligand. This caspase-dependent apoptosis can be mimicked in vitro using a strong stimulus, for example, using anti-CD3 or CD38 antibodies. Our assay involves the screening of kinase inhibitor libraries for their effects on the modulation of T cell activation markers, and this protocol would benefit from increased automation, and improvements in staining could help in the study of specific T cell subsets. For the assay, we stimulated mouse thymocytes with anti-CD3 CD28 coated beads overnight and measured the levels of caspase-3 activation and CD69 upregulation. To examine the potential benefits that the DA cell washing system could have, we set up an assay to compare the different methods of carrying out our assay. The first protocol involves the use of a standard 96 well plates for culturing the cells for the duration of cell stimulation.
Sorry.
And it's okay. And also the centrifuge dependent washing steps. For the second protocol, we used the standard 96 well plates for cell culture before transferring the cells to the DA plates and using the laminar flow washing steps. But for this second protocol, it requires at least one centrifugation step where we transfer the cells from the 96 well plate to the DA cell plates. And lastly, our third protocol uses the cultures the cells directly in the DA cell plates and using the laminar flow washing steps. This last protocol will eliminate any centrifugation steps compared to the other two protocols. So moving on to our results, comparing the results of our different protocols, they give relatively similar staining across the different samples. However, we do see higher MFI levels for the centrifugation independent protocols. So looking at the columns 2 and 3, comparing them to the plots from column 1. So in the non-stimulated samples, the active caspase-3 levels represent the background activation of caspase-3, while the dexamethasone treated samples are the positive controls for caspase-3 activation. And in our stimulated and DMSO mock treated samples, in our next slide, please.
Okay, sorry.
In our stimulated and DMSO mock treated samples, our caspase-3 activation was observed, which is a result of stimulation-induced apoptosis. And in one of our inhibitor-treated samples, in this case, the inhibitor PIK75, the caspase-3 levels were suppressed, indicating that this inhibitor does inhibit T cell activation. And once again, the MFI levels for the centrifugation-independent protocols were higher than the centrifugation-dependent ones. Another activation marker that we looked at was CD69. For the non-stimulated and dexamethasone treated samples, CD69 was not expected to be upregulated in this case, as they were not stimulated. So for the stimulated samples, CD69 levels were upregulated, and again, the treatment with PIK75 suppressed this upregulation of CD69. So to summarize our results for the first half of our presentation, the DA cell washer is useful for helping to improve staining with certain critical antibodies, and this can help us to gauge on relative populations better. And also, in the case of our inhibitor screen, the DA cell washer has helped to improve the efficiency of carrying out our screening assays, and the automation of this process helped to produce consistent results, and it's really useful for such high throughput assays. Critically, our results.
Thank you, Elijah. Sorry.
Yeah.
Okay, thank you, Elijah. And I'm going to continue the story and show you some data that we have on cytokine signaling in mature peripheral T cells. So with those experiments, we had two problems, one of which was the cytokine receptor expression after T cell activation, in which some of our antibodies were giving us very problematic staining, as you will see. And another issue we had, we tried to develop an experimental system to allow us to examine a duration of the cytokine signal after the cytokine itself has been removed. And for this, we need very fast and actually relatively gentle washing procedure. Well, so we'll start with some very bad data. And this is an experiment in which we are looking at the expression of CD25, which is a part of the high affinity IL-2 receptor. And this gets upregulated after T cell activation. So if you examine cell trace violet dilution indicating cell proliferation, you can see that the cells that have proliferated, the cells have activated, should upregulate CD25. But you also immediately notice that with the conventional sample prep, we are getting this very problematic staining, which means our MFI analysis is probably not going to be very meaningful. And luckily for us, we were able to completely eliminate this very problematic staining by simply using the DA washer. So this actually shows you exactly the same cells, the same experiment, the same antibody. The only difference is the sample prep. So we are actually able to generate much better data using the DA sample washer. And the next application is probably going to be more interesting, because in this we are not using the DA plate washer for the antibody staining. We are using it actually during our experiment. So what we are interested in this experiment is we want to test how long will IL-7 induce STAT5 phosphorylation, how long will that persist after the IL-7 has been removed? So we are looking at ex vivo freshly isolated T cells. We stimulate them with IL-7. This will induce the STAT5 phosphorylation. And after this step, we wash off the cells to remove IL-7 and fix our cells and follow by methanol permeabilization at different time points to allow us to examine the signal decay. And for this, we compared both conventional centrifuge-based approach and the DA plate washer-based approach. So we are starting with exactly the same cells with the same 10-minute stimulation. With the conventional centrifuge, we did 3 times 5-minute conventional centrifugation using a 15 ml wash volume. And we really wanted to use, as many centrifugation steps as possible, because T cells are extremely sensitive to IL-7, and we really wanted to make sure that we will be able to actually remove IL-7 completely. And after the centrifugation, the cells were either immediately fixed with paraformaldehyde, or they were incubated in IL-7 free media for the given amount of time, followed by PFA fixation. So in this case, we have 3 washes, and our approximate time is, well, around 20 minutes. Whereas with the DA plate washer-based approach, we did 9 washes, which took us approximately 2.5 minutes. So you can see we're using the DA plate washers, we are getting like 3 times as many washes in like 1/10 of the time. But is it helping us with the experiment? And I believe the answer is yes, because if you look at the data, and in this experiment we are looking at CD8 T cells. So if we just look at the STAT5 phosphorylation directly 10 minutes after activation, we see, relatively comparable STAT5 phosphorylation level using the two different sample prep methods. However, if we try to look at the kinetics of the signal decay, so after the washes, we put back the cells for half an hour or hour and a half into IL-7-free media, and at the end of each of those time points we permeabilize, it's quite obvious that we see relatively little of the signal decay using the conventional sample prep. However, there is a very significant decay of our STAT5 phosphorylation if the samples were treated using the DA plate washer. So with the conventional sample prep, it really takes at least hour and a half to see any of the signal decay, whereas with the DA washer sample prep, our signal decay decays actually within the first half an hour. And we observe exactly the same effect when we're looking at the IL-7 signal in CD4 T cells, where you see very little of the signal decay within the first half an hour after the IL-7 removal, whereas the signal decreases very significantly within the first half an hour when we use the DA plate washer for the sample prep. And we think that the main reason for this difference in the kinetics is that the T cells are very sensitive to IL-7. And because we have more washes with the DA plate washer, we are just removing the IL-7 much more efficiently. And we think that this is actually the real biology. And what we see with the conventional sample prep is an experimental artifact. So in summary, we are using a lot of flow cytometry to look at the TCR and cytokine receptor signaling. And for our experiments looking at thymocyte development, we get significantly better staining intensity for the thymocytes using several critical antibodies. We have used the DA plate washer to develop a very consistent and time-efficient high-throughput thymocyte activation assay. And for our experiments on cytokine signaling in mature T cells, definitely the use of plate washer allowed us to remove problems with some of the antibodies, as well as it actually allowed us to do some meaningful biology to examine the duration of STAT5 phosphorylation after removal of IL-7. So thank you for your attention, and we will be happy to take any questions. Thank you.
Wonderful. Thank you so much, Joanna and Elijah. That was an excellent presentation. If you have any questions, please type them in the question box, but we do have a few that have come in. The first one, in the slide of IL-7 stimulation and washing, in the condition of no IL-7, there seems to be a shoulder at the far left condition for DA, while not in conventional method. Can you explain why this happens and what it is?
Okay, so that's a very good question, and I'm not 100% convinced this is a real shoulder. Okay, so we are comparing this with this, and I think this is, in both cases, probably a bit of experimental artifact. And we have this shoulder even in the conventional sample prep. It may be less obvious, which may be actually related to the fact that, our total MFI is higher on the DA sample prep. So I think this kind of shoulder is either coming from incomplete permeabilization. It could be actually coming from cells that express very low levels of IL-7 receptor. And in this experiment, we are not gating on subpopulations within either CD8 or CD4. So for example, if you look at the CD4s, the shoulder is probably more evident, but in this experiment, we are actually not gating out the Treg population. And the Tregs are supposed to have extremely low level of the IL-7 receptor expression. So I think, what looks like a more like a shoulder in the DA plate washer is just a result of, higher staining intensity. Saying this, it's a good question. We can probably try to look at the data more carefully, try to see what's going on there.
Great, thank you. Another question. Why do you think antibody staining is stronger on DA plate than conventional plates? Isn't it using the same antibody concentration and incubation time?
So it is using exactly the same antibody. So in those experiments, we're using, absolutely the same antibodies. And the samples in all the experiments I'm showing you, this is exactly the same cells, and everything was processed in parallel. So I think, having, better washes probably helps, not centrifuging the cells probably helps. I think, the cells are just, I know you retain, better membrane integrity. I think, the curious people may be able to give you more of the more technical answer. All I can say is it's working. And there is one more thing. In some cases you do actually get lower MFI if you use the D8 plate washer, such like in this pretty horrible staining. This is exactly the same antibody, exactly the same cells, so you can actually get lower MFI with the DA washer sample prep, but this is definitely a real staining, unlike this one.
Thank you. Another question. How was transferring the assay from a conventional plate to the DA plate? How long did it take? And what did you have to change, if anything, before you saw comparable data on a DA plate?
Okay, so the biggest difference, and the only thing that really needs optimizing, is you are using lower volumes for the DA washer sample prep, and which means that you need to, test. Well, can you use the same antibody concentration at a lower at a lower volume. So it didn't actually take us that much to do the optimization, at least for the antibody staining, because we got some help from the curious people in terms of how we can transfer our conventional staining for a plate-based staining. And we did one pilot experiment. It worked extremely well, and that was it. I think Elijah can probably tell you more about optimizing with doing the stimulation, the plate itself. But I think even that was relatively straightforward. So it's like you optimize doing one experiment.
Thank you. One last question I see. Is there truly no mixing between the wells?
I really think so, because we would have noticed that. And you can actually, so after each wash, you can actually look at your plate, and it looks really clean. We have used some very different cell types on the same plate, and we would definitely notice if we got mixing of between wells, and so far it has not happened. So it is one of those things. It does look like, well, it's magic, it's impossible, but so far we have never experienced that, and I believe it just doesn't happen. I think it's more likely we'll mix something by mistake when we are doing the conventional sample prep and manually pipetting for the washes. So I'm actually confident there is no sample mixing.
Wonderful. Okay, that was our last question. Thank you so much to Joanna and Elijah. Really appreciate it. Thank you everyone for attending.